Usually it says recording in progress, but it says it's recording.
Oh, okay, welcome everybody. It's my pleasure today to introduce Dr. Heather Gibson. I've worked with Heather really over the last couple of years in a project that she takes the lead in, and she's gonna student. Dr. Gibson got her PhD here at Wayne State in immunology, correct?
Mhm.
She did her, in fact, and you stayed here.
Yeah, love it here.
Good decision. Right, so she did her postdoctoral work, moved up the ranks, assistant professor. She is an associate professor with tenure now, this year. Congratulations to you.
Thank you.
Again, well deserved. And today she's going to talk about a unique model, a very powerful model, genetically diverse mice to study immune regulatory mechanisms. Dr. Gibson.
Thank you, Dr. Stemmer. Well, not really good. Okay, yeah, just make sure you actually want to clap based off of what I'm going to show today. So I invite you guys to just interrupt me if you have questions along the way. And like I said, if you see anything on the chat, because I don't have the thing up on here anymore, just go ahead and stop me because I don't want to lose you at the beginning and have you not understand any of the rest. There's a couple of different blocks of what I'm going to talk about today. This is going to be largely focused on, I was awarded the CURES Recruited Center Investigator Award for 2025. So that came with some funding to help boost this project, and we're hoping to get this to evolve into something that can be R1 level funding. But most of what I do is actually in cancer immunology. So this is a little bit of a pivot from our normal day-to-day, but we're applying these same genetically diverse mouse models to study some of these immune regulatory mechanisms to mercury as a toxin. Okay, so hopefully we're all aware that mercury is dangerous. Exposure can lead to multiple different health effects that can affect multiple different systems within the body. There's lots of different sources of mercury that are out there. It can be either inhaled, ingested, or contact with the skin. And so today we're going to focus on its effects within the immune system. And so some of these additional notes on where you could be come into contact with mercury through the soil, through the atmosphere. So this particularly affects vulnerable populations that are living in industrial areas. Coal-fired power stations, incinerators can release mercury into the air, and the exposure to that can actually increase this as a risk and a disparity within our community. Also within the water, which also then affects aquatic life. So food that you would be ingesting, dietary consumption within fish predominantly get a bad rap for having high levels of mercury. But in general, this continues to creep into all of our diets through some of these water contaminants as well. Then there's also some examples of medical treatments and cosmetics. Preservatives, low levels of mercury used to be used in multi-dose vials of the influenza vaccine, and also for other vaccines, which came under fire for a potential link to autism. We can get into that discussion later. I'm not going to go into any of that today, but occupational exposure, of course, if you're working in any of these different regions where you'd have a coal plant or other incinerators, and then other various types of jobs that utilize mercury for what they're doing. And then some medical and scientific instruments also could actually end up causing you to have an exposure to the mercury. So like I mentioned today, we're going to talk mostly about the immunologic effects. And hopefully by the end, I'll have you convinced that there is some genetic susceptibility in individual genotypes that might predispose. And so the goal overall of these studies is to figure out what genetically distinguishes somebody who's going to be more or less sensitive from the immunological aspect strictly. And we're using, genetically diverse mouse models for that. So, this work has been going on outside of, when I got involved for about 100 years. We've known that mercury is toxic. We know that it can have an effect on the immune system. It can predispose patients to autoimmune conditions. It can increase the severity of autoimmune conditions in people. And so, what was done about 20 years ago, this paper was published, showing various different strains. This H2 haplotype is the MHC subset. So MHC locus, or HLA is what we call it in humans, is the most polymorphic locus in our genome. this is the region where you have proteins that are actually presenting antigens to your T cells. So it's very important for getting an adaptive immune response activated. And so, in this case, they were looking at different H2 haplotypes in these different backgrounds. So you can see there's just differences here. We don't need to know exactly what these are functionally doing. But there seems to be like this S haplotype seems to be more associated with renal immunoglobulin deposits. So this is something that would be like a lupus-like situation where you have antibodies that are creating these large clusters. And then as a result, they deposit within the kidneys and cause damage there. This AFA was just their particular autoantibody of choice. This was an anti-fibrillary antibody. And you can see that this seems to be skewed towards this S haplotype here. And then in the case of like the renal accumulation and retention, there was some variability based off of the genetic backgrounds. And so, this was certainly proof to me that there was a chance that we could possibly use some of the outbred models that I've worked with previously to try to study what is it outside of, this HLA type, what other genes might be associated with the toxicities that happen with exposure to mercury. Another figure from this paper, or table from this paper, was also showing, increased concentrations of mercury dose. Just, for the record, 5 to 10 mg per liter is roughly what would be considered about 1 part per billion, which would be accumulation within the body over long-term chronic exposure. This falls below the FDA guidelines. Anything over 2 parts per billion is considered like a red flag. So these are levels of mercury that we might all be exposed to that, if we continue to have an EPA, which I hope we do, this is like, below that threshold as well. So not to make everybody panic, but, these are like real life situations that we're all dealing with that fall underneath that EPA radar. So again, they're separating this by these MHC haplotypes and, again, looking at this autoantibody and then, the renal mercury retention. And you can see that there are variations here based off of the genetic backgrounds. And one that I think is important here is even with this, S haplotype of the H2 locus, you can see that there are differences in the titer of these autoantibodies that are being produced between these two that are both at S haplotype, and then also the retention within the kidneys. So again, arguing that there's probably more than just what's happening within that MHC locus. So aside from this, what genes are regulating mercury retention, and also that induction of those autoimmune phenotypes. And so I show this slide pretty much every time I talk about the genetic linkage. This was a really nice review article that looked at outside of MHC, what genes seem to have linkage to autoimmunity. So we've got a whole bunch of different autoimmune diseases, diseases shown here. And then in between them, we've got some genes that are either in black if there are polymorphisms in there that are shared with 2 autoimmune disorders, or if they're in red, there's more than 2 autoimmune disorders that share polymorphisms in these genes. And so these aren't mutations, these are just normal single nucleotide polymorphisms that are different between each of us that make us genetically unique, that might predispose us to, either more likelihood of getting an autoimmune disease or the severity of that autoimmune disease being higher. So, how do we study this in mouse models? We often work with inbred mice, which is great if you're trying to control for the genetics of the mouse. But in our case, we're trying to actually use our variable being genetics. So I was quite fortunate that a lot of people put a lot of work in well before I started working with this model back in 2002. A group of geneticists at this complex trait consortium proposed to generate these recombinant inbred strains from 8 different founders using mouse musculus, mouse domesticus, and mouse castaneus, which is just one, the cast background. I highlight here some of the people that I've actually met along the way of working with this model, and this was back in 2004, so over 2 decades ago. They published this, and their idea was that they were going to make these like just a whole bunch of recombinant inbred strains based off of these 8 founder strains for people to be able to use. And so, this little figure here summarizes, I think, quite well what we're working with. So these are the different eight inbred strains that were crossbred to create these outbred models. They refer to these over on the left. Some of these might look familiar, like 129, C57 Black 6 is B6, AJ. People work with these generally, pretty reasonably high levels of these being used in laboratories. And then these over here are a little bit newer to the scene. They refer to them as wild-derived strains, which I find kind of funny. None of these were made in a petri dish. All of these were at some point somebody caught a mouse, started inbreeding it, and then they selected for the ones that were probably more docile and able to be bred. And so then as a result, this is how we created all of these different inbred strains. But these 3 off to the right here are a little bit newer to our research use. So what they've done here is there's two different directions that these breeding funnels can go. The original intent was to create this collaborative cross recombinant inbred. So what does that mean? They did about 25 to 35 generations of breeding to cross these 8 inbred founder strains, and then they re-inbred them, thinking they were going to get thousands and thousands of different strains to work with. It turns out there's about 68 available at University of North Carolina. It's been 20 years that they've been working on this. So what does that tell us? It is not advantageous to be inbred. We already knew that. But anyway, so at least though there are 68 strains that are available there, hopefully they'll be able to maintain those. Every once in a while, another one drops off the list because they can't maintain the breeding. But the idea here is that you've got mice that have mixed genetics that are representing each of these different founders. And then you also have them as an inbred population, so you can actually buy more of them and do more experiments with them. Alternatively, on the right side here is the diversity outbred. So these are just continuously outbred. They're in like their 85th generation of outbreeding at this point. These are maintained at Jackson Labs. They do all the breeding there. You can purchase them in different waves. So once they've got one available, they post the dates of when you need to put the order in. You can purchase these mice directly from Jackson Labs. So you don't have to do any of that outbreeding on your own. That would be just a giant disaster. So the idea here, though, is that each one of these mice is genetically unique. Yes, question.
Have they maintained the genetic diversity?
Oh, absolutely. Yes. So the way that they're breeding them is not sibling mating. So they're just like breeding DO with DO with DO. And so, yeah, 100%. I have asked them periodically. I'll get to this idea of haplotype blocks in a few seconds here. But as they continue to outbreed, the haplotype blocks are going to get smaller and smaller and smaller, which could become problematic later on. But for right now, yes, they maintain all that genetic diversity. Because they are not homozygous at every locus. So that is the downside to being inbred, is you're homozygous at every locus. You've got 2 copies of every chromosome, right? And so if one of your copies has something bad in it that's not advantageous for you to be alive, you hopefully have another copy that has something in there that's going to counter that. In the case of being inbred, if you've got 2 copies of the bad thing, then you don't survive. So that's why it's so difficult to create these inbred strains in the first place. But with the outbred situation, they're heterozygous everywhere, or could be homozygous, at the same rate that we would probably be as humans. But yeah, so definitely lots of genetic diversity that are in these. And as mentioned, heterozygosity is present. The idea behind picking these 8 inbred strains was they wanted to, I keep hitting the side button, cover about 90% of all the genetic diversity that's known in mice. Also, all of these have been fully sequenced, which is going to become important for when we try to do the genetic linkage analysis. So we know what the entire genome is of all of these 8 founder strains. We have tools that we can use to piece back together the genomes of every one of these DO mice if you work with them. And then as a result, you can use these genetic mapping tools that you can do these genome-wide association studies where you're linking the genetics to your phenotype of interest. So this is an example of one of the mice that Carl Broman sent me this plot when we were trying to figure out how does this work. This is the guy who actually wrote the code in the R platform QTL2. He, as of a couple of years ago, was still personally responsive to emails and helping us work through our data analysis, which is kind of rare. So it's still an exciting time to get involved in this. And if you think about trying to apply this model to anything that you're working on, and if I can help you in any way, please reach out. I'm happy to help you because I think more people should be using this. It's super powerful. So as mentioned, they were shown kind of here in very tiny little pictographs, but this is just a zoomed in all the different chromosomes of an individual DO mouse. So you can see that there's a lot of heterozygosity here, some homozygosity though, just like we would have in humans, and every single one of your mice is going to be a unique mosaic of these original 8 founder strains. And so when I mentioned the haplotype blocks, that's where it's like this whole thing here is from this cast background. That block is a haplotype block that's coming from cast. As you continue to do this outbreeding, those haplotype blocks are going to get shorter and shorter because there's going to be meiotic recombination sites, chiasma that are present in there. So eventually it might become problematic for doing the actual genotyping, but so far we're okay on that. And most of you probably don't know what I'm talking about with that anyway. So hopefully it continues to not be a problem. So, all right, so how diverse are these? This was something that I did back when I was a postdoc. So this is probably over 10 years old now at this point, and I did it again. So just looking at the peripheral blood content. So in humans, this is the range of B cells, T cells, CD4 T cells, regulatory T cells that we would expect to find in the peripheral blood of a human. In the DO population, these are the ranges. And so some things might stick out to you that like, wow, mice have a lot more B cells than humans do in their blood. There are some differences between humans and mice in terms of blood content. We have a lot more monocytes and neutrophils than mice do. They mostly have B cells. But what I would hope that you would appreciate here is the distribution of the percentage of the B cells, percentages of the T cells, and I have for comparison here the B6 background and the BALB/C background, where they're very tight. So genetically, there seem to be relationships between whether or not you have more or fewer B cells and T cells, and then even the distributions of the CD4 helper T cells versus CD8 cytotoxic T cells. And then within that CD4 subset, FOXP3 is a marker of regulatory T cells, and there's a wide range here, whereas in the inbred populations, it's very tight. There are actually people who've done genetic linkage to this, and some of those reports are published. So if you're interested in that, I'm sure you can find it in the literature. So this is, I think, just a really nice representation, though, of the amount of diversity and how this might relate to immune function in these mouse models. Also, just generally who's using these mice and why, and selfishly I put 3 of my own papers on here, but there's a lot of people working in toxicology, obviously immunity, cancer, and in physiology. And so this is, I should have put the image of how many citations have included diversity outbred in there, because over the years it's continued to grow. It's kind of stabilized out at this point, but there's quite a few papers every year coming out where they've utilized either this or the collaborative cross in order to be able to study the genetic linkage of various different features. I often get asked, why not just do this in humans? There's a couple of different reasons on the cancer side of things. You can't control for tumor heterogeneity very well in humans, so we use these as a way to get around that. But for most of today's purposes, that's irrelevant. But one other nice feature is that something you need to be aware of when you're doing genetic linkage is the frequency of the minor allele. So that polymorphism that's making it stick out, if that's less than 50% of the population has that particular nucleotide in that spot, that's the minor allele. And generally, in order to have enough of an N to power your study, you want that minor allele frequency to be at least 5%. So you can see over here on the right, now this data is kind of old because they've continued to outbreed them, so I'm sure this minor allele frequency is going to change over time. But this is as of about 10 years ago. Dan Gotti gave me this. He used to work at Jackson Labs, helping to maintain these models. And it's basically almost all of the polymorphisms in the DO background are above that threshold of 5% minor allele frequency, whereas in humans, most of these minor alleles are not above that. They're even below 1%. So if you look at the 1,000 Genomes Project, there's about 85 million reported SNPs in that project. Only about 8.2 million of those reach that threshold or above that threshold, whereas in the DO population, there's about 40 million SNPs. Almost all of them are above the 5%. So it's a great model for powering your study. And so this is some power analysis that we're showing over here. Due to those increases in the minor allele frequencies, unlike the human studies, you don't need generally thousands of subjects. There's some things that can contribute to whether or not you need more or fewer. You can generally work with somewhere on the order of 200 to 400. I've often gotten away with 100 to 150 for the studies that I've done. And I'll show you a little bit of data for the cancer stuff just so you can see how this all applies to the big picture. Nice thing is you can control external variables as well when you're working with these mice. In terms of mercury, you can treat them and dose them on the same schedule. Humans are exposed to mercury at different rates, and so that also throws in an additional caveat. Environmental and lifestyle factors can be largely controlled. And then in terms of our cancer work, we can control for tumor heterogeneity simply by crossing the DO mouse with the mouse where a syngeneic tumor came from. And now these mice that are the progeny, one copy of every chromosome is coming from this strain where the tumor came from. And as a result, they don't see that as foreign, so they'll accept those tumors. In the case of the mercury studies, we can actually just work directly with the DO, though. So I mentioned some difficulties that might happen as these haplotype blocks keep getting shorter. Right now, there's this low-cost genotyping array that costs about $100 per mouse. It's called GigaMuga. It's the 3rd generation, hence the Giga. But this is Mouse Universal Genotyping Array. It has a 143,259 probes on it. Most of these are SNPs. Some of them are insertions and deletions as well, but they're just genetic markers that are then trying to tell us which alleles are present there. Those alleles are identifying which founder strain contributed that block of the DNA. So it's read out as in, does this mouse only have allele 1? Is it heterozygous where it has both allele 1 and allele 2, or does it just have allele 2 for every single one of these almost 150,000 probes? So that haplotype reconstruction, for those of you who aren't well versed in genetics, I certainly was not when I first started working with this project. If I would have gone back in time to when I was a graduate student, I never would have imagined this is what I was going to be doing. But this was a unique opportunity to get involved with this project as a postdoc, working with these mice, and I saw the power of it and wanted to continue that to today. So I look for all the students in the room. What you think you're going to be doing might not be what you end up doing, but just go after the things that excite you, because if it gets you out of bed every morning, it's going to be awesome for you to work on it in the future. So all of these X's on here are what we call recombination chiasma. These are meiotic homologous locations where this is where the crossing over happens in the F2 generation. So you have to cross again because this is during meiosis, not during mitosis. So we've got our DO founder, and then in our case, for most of our studies, we're crossing them with an inbred strain. So what the result is, as I mentioned, is one copy of the chromosome is coming from this B6 mouse, and then the other copy is one of the two from that DO. So then as a result, the point of all of this is to say that that array that has almost 150,000 SNPs, there's obviously way more. I just told you there's like 40 million in there. So how am I getting any data out of 150,000? It's because that array is not just identifying a causal SNP, it's identifying the block. So now I know that this whole chunk of DNA, obviously this side was always going to be B6, but this portion here came from this light blue background that equates to the NZO background in the founder strains. And so now, because those were fully sequenced, I can impute all of the polymorphisms that are associated with NZO. And as a result, I know the entire sequence of this. Same thing for this region, and so on down the line. So you can piece together the entire genome of the mouse. And I guess I should have duplicated this part. These regions here where it's white are the questionable regions where it had a tough time because that gigamuga wasn't able to identify exactly where that block or what that block was made of. It can kind of take a guess because CAST is it's changing the speaker. Hopefully that's okay.
Yeah, I think the signal cut out for a second.
Okay.
It looks okay.
Just making sure everybody can still hear. So online, you guys can obviously still hear it's unrelated. So, but yeah, so this is something that I have a little bit of concern with as we continue to do this outbreeding. There might be more of these white regions, and they may need to come up with a Terramuga for the next round that has even more polymorphisms on it. But for right now, it still seems like it's able to do a reasonable job. And you can test that as you do your run to do your haplotype reconstruction. You can actually have it spit out that exact plot for every single one of your mice. Okay, so I want to go into how we've used the DO model previously, because a lot of people have asked, you get a lot of GWAS studies that have been out there, and where has that gotten us? Like, what have we actually learned from that? Has it ever applied to anything? It's like we found these linkages, and then it just kind of stops from there. And so I kind of want to go into some of the cancer stuff that we've done so you can get an idea of like where we plan to go with the Mercury project. So if you'll bear with me, I'm going to overcomplicate it by putting two different projects that we're working on simultaneously, side by side throughout here, but just try to follow with the basics, and I won't go into too many details. So on the left, we're looking at anti-tumor antibody. So this is something like trastuzumab, which is an antibody that's therapeutically used for HER2 overexpressing cancers. And then this on the right-hand side is immune checkpoint inhibitors. Hopefully you're familiar with those. There's commercials for them now. This has been the biggest breakthrough in cancer immunology ever, where we've actually been able to put the brakes or block the braking mechanisms that are happening between cancer and the immune system. Okay, so why do we think there's genetic linkage? Well, if we look at survival curves, so this is in clinical trials, starting out 100% of your patients are alive. You start them on their different treatments, and then you measure over time how many of them continue to survive. So this is what the survival plot is here for that exact antibody that I mentioned, trastuzumab. This is comparing it to a biosimilar, but I kind of like this plot because it's recent. Trastuzumab has been around since like the 80s. Some of the original trials might have even been in the 70s. Our clinical care is better now. So having something that's today's data with trastuzumab on its own in patients with metastatic HER2 positive breast cancer, I think, is powerful. So then over here on the right, same thing for, immune checkpoint inhibitors across the trial, either combining two different checkpoint inhibitors, PD-1 and CTLA-4, or just PD-1, just CTLA-4. This is 10-year data. And actually, in this case, they're showing melanoma specific survival because it's 10 years out. So these patients are probably cured of their disease. Some of them were dying for different reasons. So when they reported this, they had to specify that it was melanoma specific survival, which is really awesome. So in this case, all of these tumors, have some HER2 positivity to them. In this case, this is metastatic melanoma. There's a lot of mutations in melanoma, so we would think it would be super immunogenic, So why are we only getting about 50% of patients responding in both situations? Well, there's obviously a lot of reasons, but tumor heterogeneity could be one of them. Maybe we've gone after the wrong checkpoint. Maybe there's a third option we should include in here. But obviously what my lab is focused on is how does the host genetic background contribute to this? So I mentioned, a lot of people when they work with mice are working with inbred mice. And so what do you get out of that? So say we've got our model for our anti-tumor antibody therapy. This is the tumor that these mice get. Here's our version of the anti-HER2 new antibody that they're getting as their therapy. And if we dose them with this, it seems to control the tumor during the dosing, but if we give them enough time to get an adaptive immune system primed, and then we take the treatment away, those tumors come right back. And it's basically every one of the biopsy mice does that. So what is that going to tell us about the human population? Hopefully I've made a strong enough argument about the fact that these mice are both homozygous at every locus. They don't represent any human, really. On the right-hand side, immune checkpoint inhibitors, kind of similar situation where if we give them a melanoma model, a mouse melanoma model, treat them with anti-CTLA-4 and anti-PD-1, they don't respond to that treatment. So what have we learned from this? Because maybe this doesn't even really represent a human. So bringing it into using the DO and the collaborative cross mice, we worked with the diversity outbred for our discovery phase. And then once we've identified a locus of interest, we go back and then we use these collaborative cross mice selecting for the genetics of interest based off of what we found in our discovery phase with the DO. So, again, we've got, our cross here where we just create, a whole army of F1 mice that we can now give syngeneic tumors to. And then on the left-hand side here, we're looking at anti-tumor antibody therapy. About 45% of them completely eliminated the tumor, about 55% didn't. And even in these partial responders, if we plot the distribution of the tumor doubling time, there's a wide range of responses here. So this is great because this is what you want if you're going to do this genetic analysis. You want it to be something that's consistent in inbred mice because that proves that and you want there to be different inbred mice that have different responses, just like we saw with the Mercury stuff at the very beginning of this talk. But you want there to be a wide distribution if you've changed the genetics. And so that's what we were seeing here. And then the same thing was true for the immune checkpoint inhibitors. We had about 13% of the mice that completely eliminated the tumor. This is looking at onset. So how long did it take for these tumors to reach this 150 cubic millimeter threshold? This distribution is also quite wide. So this is great. This is our phenotype. This is our number that we're going to plug in when we do that genetic linkage analysis. So ideally, you would have a continuous phenotype like this that you could enter numbers if you're going to do the downstream analysis, but you can also do binary where it's either yes or no for whatever your scientific question is. And then as I mentioned, Gigamuga, it's about $100 a mouse. You use this RQTL2 that was written by Carl Broman to reconstruct the haplotypes. And then there's a whole bunch of other downstream things like associating the genomic loci with your phenotype. So what do we get when we do that? Well, this is what we're looking at here is the immune checkpoint inhibitors on the right. And again, the anti-tumor antibody on the left. There are these peaks that show up here. So this is across the entire genome of the mouse, every chromosome. How closely does that exact region of the chromosome associate with your phenotype? So the higher the peak, the stronger this logarithm of odds score. It's sort of like an inverse p-value, log transformed p-value. So these arrows are pointing at regions of interest. For our anti-tumor antibody therapy, we chose to focus on chromosome 10 because it was tough for us to figure out what the exact driver genetics were in chromosome 1. And then in the immune checkpoint inhibitors, we've looked at loci both on chromosome 13, and then there's one on chromosome 8 that we were interested in as well. But one thing I want to kind of point out is these do not look the same as each other. So each different type of immunological question we're asking is likely being driven by different features. So of course, the loci that come up are going to be different between them. And we expect the same thing for our Mercury studies once we get to the point where we can do the genetic analysis. Another interesting point that I thought was kind of fascinating is the mouse MHC locus that I talked about being super polymorphic and it can drive autoimmunity. And in a way, cancer immunology is trying to drive a very focused type of autoimmunity. That's on chromosome 17, and we don't have any significant peaks in either of these models. So perhaps this is a good opportunity to look for things that are outside of the MHC locus if these mice don't have enough differences within that locus to make it come out as a strong predictor. So now, in our lab, we're trying to test the association of this chromosome 10 locus, the chromosome 8 here, and then also chromosome 13. So one of the other features that you can do, I'm focusing on chromosome 13 because we've kind of gone the furthest with this. This is another feature of that RQTL2 platform that you can't do in humans. You can go back and actually ask which of these original founder strains was driving your phenotype. So if we start out at 0 here, anything above that is a positive regulator of the phenotype. Anything below 0 is a negative regulator. And here is our peak. If we zoom in on chromosome 13, we can see that NZO, this light blue, is a positive driver and B6 is a negative driver. So now that we can actually go back and identify collaborative cross mice that have those genotypes in those loci. So the UNC System Genetics website, and if you're interested in this, I can send you the links to all of this so you don't have to like scramble to write it down. But they've got a really nice browser on there that you can not only just look at the entire genome of each mouse, but you can also search for specific loci, and it'll tell you which of those 68 CC mice have your genetics of interest. It'll break it down by the different backgrounds that are present there. So we did that. So again, back to chromosome 13 and our locus of interest. We identified a couple of mice that had CC17, CC44 that were B6, exactly where our peak is located on chromosome 13, and then NZO as our putative positive responders. And so the idea is generally we try to have at least 3, because if we're going to validate that these genetics matter, these are all genetically different everywhere else. So having enough to sort of power this idea. We're a little bit limited by the fact that they only have 68. And then we also try to select mice that do not have confounding genetics at other loci as well, which is kind of important. Okay, so just as an example of us trying to validate, so again, sticking with chromosome 13, we do the cross with the B6 mice, give them the tumor, give them the treatment. The 2 that were predicted do not respond, do not show any response to the checkpoint inhibitor therapy, but 2 out of the 3 that we predicted to respond do show response here. So it's these 2 on the far right. That, if we just look at the genetics of chromosome 13 alone, it seems like it's driving that effect. And then we've, looked at CD8 T cell infiltrates as, a metric of, was this immunologically driven? And it does appear that there are more CD8 infiltrates in these responder mice. Okay, so the whole point of all of this isn't just to, validate the locus and create models. We wanted to know why, and can we do something about it? So zooming in on what genes live in this locus, we were lucky that there's just the prolactin gene family here. There is one additional gene, but we super focused on the prolactin locus and started to do some validation studies with that. At first I was like, prolactin, that's involved in lactation. There's no way that's involved in immune response. And I had an MD PhD student who was brand new in the lab. My lab was brand new at the time as well. So he was trying to write his F grant, and I was like, I just don't know if anybody's going to believe us if we say prolactin is driving an immune response. Maybe pick a different locus, which is where we started working on chromosome 8. And then I just Googled prolactin and immune response, and it turns out there are many, many decades. In fact, there's one of the original papers that came out was about 50 years ago in the Lancet, titled something like Prolactin and Immune Regulation. So not something that they teach you. I did get my PhD here in immunology, but other immunologists that were trained elsewhere also were not aware of this. So yeah, question?
I just wanted to make a comment that this is where actually AI is really useful in doing these kinds of searches to try to pull together disparate literature.
Yes, absolutely.
Stuff like
You have to be careful and go back and check
Yes.
and make sure that they're not just making stuff up, because AI is also really good at making things up. But
Yes.
it does give a really nice summary sometimes in that AI summary when you search using various different search engines. And so this happened to be Google because that was the most popular at the time. But yeah, there's a lot of immune functions that prolactin can have. In fact, it's also linked to autoimmunity, similar to what we're working with Mercury. Mercury has a tendency to cause a lupus-like effect in people. High levels of prolactin, not the height of lactation. So lactation is like 1,000-fold higher than normal. We're looking at like double what the normal levels would be. Can actually be immune stimulating. You get to be too high, and of course in immunology, everything has to be this delicate balance. You get to be too high, it's actually immune suppressive. So kind of a fun thing to study because it's also has a circadian rhythm to it as well. So my poor students having to deal with this, but yeah, so it does look like maybe there is actual justification for looking at prolactin. So we tried supplementing mice with prolactin itself. And then my new MD PhD student was like, well, why not, instead of using prolactin alone, because it's predominantly produced by the pituitary gland. It has a very short in vivo circulating half-life though. And it's probably not something that patients are going to want to be put on. I have a feeling mostly men would be like, why are you giving me prolactin as part of my cancer therapy? So instead, we decided to work with what is already known. It's produced by the pituitary gland. It's regulated by the dopamine receptors in the pituitary gland. So perhaps maybe we could antagonize those dopamine receptors to then increase the levels of circulating prolactin. So here's just a little vignette too of the circadian rhythm of prolactin, which then means that it peaks in the evening and then starts to wane as we wake up because dopamine levels go down as we're asleep. and then come back on as we're awake. So there's that regulation of like the light cycles based off of the dopamine that regulates the prolactin levels under normal homeostatic conditions. So we asked, can we manipulate using this drug metoclopramide that again is going to be antagonizing the dopamine receptor to then increase the prolactin circulation. And what we found, interestingly, in this mouse that doesn't normally respond to checkpoint inhibitors alone, is if you just add metoclopramide, you actually get a survival benefit. And there's also an increase in CD8 T cells within that tumor. And this is on a log scale, so it might not look super impressive, but like multiply each of these by, tens. And you can see that there's like quite a few more CD8 T cells that are present in there. Then we also tested this by just breaking down our checkpoint inhibitor into the individual single arms, and also tested this in a second tumor model. And we actually see an even broader effect happening here. And it seems like it's driven predominantly by the anti-CTLA-4. So adding this experiment in, we know the response isn't just specific to that B16 melanoma that we were originally working with. And it seems like maybe it's working through CTLA-4. So this is something, metoclopramide is something that's FDA approved. This would be very easy to introduce a clinical trial to hopefully increase the response rates to the immune checkpoint inhibitors. So this is something that we're actively pursuing. So this locus itself hopefully is serving as an example of how you can use genetic linkage analysis to identify new therapeutic strategies. So far, in addition to that prolactin locus and the checkpoint inhibitors, keep hitting that button. The candidate gene from chromosome 8, we've identified HMGB2. It seems like it might be involved in the progression of T cell exhaustion. So we have a whole project dedicated to that. And then back to that anti-tumor antibody, the left side of everything that I was showing up to this point, we found SNAX3, which is our candidate gene there, and it might be involved in antigen cross presentation, which is something that might be regulating whether or not CD8 T cells get activated. So another active area of study in the lab. So hopefully we'll prove some of those naysayers wrong at some point. But this talk was supposed to be about Mercury. So we'll get back on track here. So, okay, now we're going to talk about, the different signaling mechanisms within the B and T cells and where we think we can, make an impact with our Mercury studies using these DO mice. So there's a lot of phosphorylation happens once the B cell receptor gets activated, but one of these early steps is this protein called psych. And this gets phosphorylated, and then as this whole downstream mechanism, this doesn't even summarize everything that's going on here. There's a lot of downstream signaling that happens after that. But we can measure whether or not these things are activated simply by looking for phosphorylation of these individual proteins. What we can lean on is prior studies that were done by Al Rosenspire and Paul Stemmer, and a bunch of other people as well. PubMed IDs are down here if you're interested. Just looking at like changes in phosphoserine, threonine, and tyrosine levels on amino acids with different concentrations of mercury. Now, 5 micromolar seems like that mimics pretty well what you would get exposed to in like sort of a chronic setting in vivo. So most of the stuff that we'll be working with works with the 5 micromolar, but you can see that there's differences in whether or not there are increases or decreases in these phosphoproteins just based off of the concentrations of the mercury. And then this is in B cells specifically, but if we look at psych, we see that at 5 micromolar, that's where you start to see a significant increase in that phosphorylation. It continues to go up from there. But again, this is the physiologic relevance. So if we compare, volcano plot of, the different proteins that are being upregulated or phosphorylated under the conditions of 5 micromolar of mercury, you can see there's quite a few that reach significance threshold. And if we put all of these together into a neighborhood plot to see who's related to who, and we zoom in on this box here, you can see psych is one of the proteins in here. Lin is another B cell signaling protein. There's quite a few different things that can be affected by mercury. So it's a very complex network. Interestingly, some of these proteins have also been identified to be associated with autoimmunity, and we're seeing phosphorylation of them. So if there's one asterisk next to it, it's something that's related to B cell receptor signaling. 2 would be something that is actually changing with mercury levels that are at or below 5 micromolar. So something that would be within that physiologic range. And then I'm more of a T cell person myself. I'm excited to do stuff with B cells, but selfishly, I also kind of want to look at T cell stuff. So we're also going to use ZAP70 as our sort of pseudo stand-in for whether or not we're getting T cell activation happening here. So very similar to the B cell receptor, once it gets interacting with its antigen presenting on MHC. This is CD8 T cells specifically, but CD4s have a very similar mechanism. ZAP70 starts to accumulate near that T cell receptor internal domain and then becomes phosphorylated. So we can look for ZAP70 as something that's going to indicate that those T cells are being stimulated. So when we first started this, Paul said we've been working on this for a couple of years now. We had this grand idea that, okay, we're just going to take a whole bunch of DO mice, we're going to treat them with mercury, and then we're going to see if the B cells and the T cells have different signaling. And hopefully by the tone of my voice, you'll recognize that we had to pivot, but I'll get into one. So, this was our whole grand plan. This was actually a figure from a grant that we had submitted. Then we're going to take all these DO mice, we're going to give them mercury, we're going to look for, B cell, T cell differences, do that gigamuga SNP analysis, and then basically look for association to various different signaling pathways, and then spit out, these interactions that are happening with the mercury. So we tested a few things to start out with. We had to figure out the dosage, route of delivery. This was kind of a headache, figuring out how to give the mercury to the mice. Turns out just giving it to them in the water bottle is probably the best method, which is duh, like in hindsight, that should have been what we started with, because that's generally how people are consuming it, is by ingesting it. And then we had to work out some conditions with cell isolation protocols. So that took us a little while. But then one of the things we noticed is as we were collecting the samples, we took pieces of spleen and we sent them to the Lumigen Instrument Center and had them tell us, how much mercury is contained here. Because we were just wondering, if you do this in BALB/C mice, like I said, we're trying to get to that 1 part per billion, sweet spot. The doses of mercury in BALB/C, it's pretty straightforward to get to that. It's about 10 mg per liter of mercury in their water bottles. These are all pretty consistent, but the thing that was striking to us is this is in the spleen. How much mercury is retained within that spleen is highly different based off of the DO mice. This was sort of like an, maybe doing this in vivo isn't really the best idea. Paul went on further to look also at the kidneys, which is a major excretion pathway. You notice there was a lot of renal function, things that I talked about earlier. Just looking at 5 mercury treated BALB/C versus 5 untreated, and seeing that, metallothionein comes up as something that's more highly expressed in the proteomics analysis. Whereas there's downregulation of these pancreatic enzymes, which is sort of a head scratcher that we look forward to trying to dig into a little bit further. But basically what this is telling us is that perhaps there are actually genetic linkage differences to the retention and or the clearance that's happening by genetic background, which opens up an entire different arm of study than what we were originally trying to do. So we're putting a pin in that. That's something that we will propose in future grants, but we realize if we really want to just look at B cell and T cell signaling, maybe we should just work with the B cells and the T cells, expose those to mercury, and then see what happens. So we take our DO mice, we collect and dissociate their spleens, we enrich for B cells and or T cells, and then we take these in culture and either give them mercury or no mercury. And then we've also done stimulation or no stimulation to do comparisons. So now we're looking for effects on the signaling. And then eventually the plan is to run genetic linkage analysis once we have a large enough N of what we've worked with. So again, having to figure out how to do all of this in the laboratory. Most of this is going to be done by phosphoproteomics with this wonderful new instrument that Paul is getting that we calculate 180 times better than the previous method, just by it's 5 times better at this, 5 times better at that. But we look forward to, stockpiling a whole bunch of samples. And then I think you said January, it's going to be here, December, it's going to be here. So December 3rd, we're going to run all of these because you got to get it out of the box. And then we'll be able to do all this analysis on it. So you've all heard it here, December 3rd, he's going to be running my samples on the new instrument. So, sorry, Paula, I hope your sense of humor is good enough for me to throw you in the bus. So, okay, so what we did was we started with a cell line. That's usually the easiest way to go. This came from a BALB/C background, so these are called WeHi. It's named after the people who made the cell line. I think it was out of Australia, so maybe it should be pronounced with an Australian accent. But anyway, so you take the cells, you incubate them for 10 minutes with mercury, and this is something that's well established in the literature. Al Rosenspire, who is now retired, used to do this a lot. So I learned everything I know about mercury treatments from him. He was a fantastic mentor in that regard. Then you stimulate the cells. And so we give them just 1 minute with an antibody that is to IgM. That's the basic core of the B cell receptors. So you're going to stimulate all the B cells in there, regardless of what they can recognize, simply by giving them that antibody. This is obviously B cell related, but T cells, we give them 2.5 microgram per ml of an anti-CD3, which is part of that T cell receptor complex. Again, bypassing their antigen specificity, just stimulating all of them. So when we do this, my poor research assistant has had to do so many Western blots lately to be able to get us some data for this while we try to ramp up and prove that this is worth doing the proteomics analysis in the first place. But when we do this, we see that this is the total psych protein, beta-actin for this particular blot. And then on a separate blot, we're looking at phospho psych in this case. And you can see hopefully that, with no stimulation, no mercury, this is the level of phosphorylation, maybe a little bit of an increase. And in fact, it's graphed here, a little bit of an increase with just mercury alone, without even stimulating these cells. And then, they're pretty similar once maybe a hint of an increase in this particular cell line once you've combined stimulation with the mercury. But this is kind of what we're going for here. So this was our set the stage, and then let's move from there. So now we're just going to go through Western blot after Western blot. And I kind of felt like it was necessary for me to show you all of these so that Savannah gets some credit for what she's been doing lately. She also said maybe she'll buy a cowboy hat for all the Westerns she's been doing. So hopefully you can appreciate we've got B cells and T cells separated here, the total protein and then the phospho versions of each. And, I'll just kind of flip through these, one after another. You can see in some cases, maybe there isn't much of a difference here. Then we've got some examples where there is an increase if you include mercury with the stimulation for the B cells and in the T cells in this situation. But what we kind of noticed was a trend that there is no trend. And that's kind of my favorite thing to study, is that in some cases the mercury alone is enough, but you add stimulation with the mercury and there is no change. This is the exact scenario for doing the genetic analysis. So kind of, I think, a great model for this. And then, now that you've suffered through the Westerns, this is graphically, once we go through and we normalize everything to its individual beta actin. So sort of like if you've done qPCR analysis, this is like a 2 to the negative delta delta CT. Yeah.
So,
Why not just do the phospho?
No. The signaling pathways, they should be the same in the B cells and the T cells.
They're not.
And
Oh, I see what you mean. Like the general trend of what would be happening, or you're
yes,
saying.
you're not seeing the phosphorylation changes in the T cells that you're seeing in the B cells.
Correct. Yes, and we think we have an explanation for that. Yeah, that's a great question. I'm glad you caught on to that. So this is looking at the B cells. Most of them are responsive. So if you look at just the blue, responsive to the IgM, but we're even seeing differences there. So this is maybe something that's fundamentally, how easy is it to stimulate B cells? I mean, we all are genetically unique. I think when we all started getting COVID vaccines or even just getting COVID, we realized how unique we are at the immunologic level, because some people had terrible symptoms and some people didn't even notice they were sick. So it's sort of similar to that. I like to use, that's my one COVID silver lining, is that I can use that as a representative example of how different each and every one of us are. These mice are all genetically unique as well, and their responsiveness just to baseline stimulation of B cells is very different. And then if you add in mercury, you can see that some of them, the mercury alone is enough to get stimulation to happen, but others it's not, or it's not that impressive. So this is exactly what we're going to put in as our phenotype analysis when we do that genetic linkage, and hopefully we'll be able to tease out some of the mechanisms that are involved in this. But DO number 6 seems to be our front runner for combination of stimulation with mercury. There was the greatest difference here. Now, Western blots are only semi-quantitative, so we're not, putting too much interest into this, but this is really setting the stage for preliminary data for us to argue doing the proteomics analysis. And then that will also give us the opportunity to look for more than just the phosphosite. We're going to look at every single one of those downstream molecules and look at the phosphorylation status of each of those. For the T cells, as you correctly pointed out, we barely saw any stimulation. And in a couple that we did, sometimes it seemed like the mercury did something. When you combined it with stimulation here, it didn't, but mercury on its own had an effect. So definitely striking differences by genetic background, and that's exactly what we're looking for here. So my point, yeah, you brought up an excellent point. Why are the T cells not getting stimulated at all? Well, T cells are actually way more highly regulated than B cells. In vivo, they have a lot more responsibility for being correct. So we have to make sure that they're sure. And some T cells will actually require having co-stimulation or additional cytokines that are in the periphery to influence those downstream signaling and the activation of them. So we suspect that that might be playing a role here. We can consider adding a co-stimulatory molecule into the mix, but I almost think it's kind of interesting that we have such varied differences here, that perhaps we might just leave it with the stimulation context that we have right now. We do have models to test the strength of the antigenicity as well. So I had shown this side of this previously, the downstream phosphorylation that happens if you have a high affinity peptide, so a very strong antigen, something that's foreign. So we'll use COVID as an example. If you're going to get a T cell excited about it, that's there's viral proteins in there. Those should never be present in our bodies. Our T cells are educated to not attack our bodies. They're educated, well, we don't educate them to attack foreignness, but we're basically pushing towards responding to foreignness. So strong antigens actually have much different signaling mechanisms that happen than some of these weak, low affinity peptides. And we do actually have models to test this in vitro. It would be a little bit more complicated because we'd have to cross the DO mice with the models that allow us to study that. But that is something that we plan to propose in a future grant. Okay, so in summary, hopefully I've convinced you that mercury retention and or excretion might also be genetically regulated in addition to the signaling itself. We have prior in vivo samples that we're going to run for retention in the spleen and also in the kidney. Proteomics analysis can be used. We've got a lot of samples that are currently stored frozen that we can hopefully identify some pathways of interest there. And then, our original question, the effects of mercury on immune signaling, we suspect this is genetically regulated. We hope to identify genes and pathways that are involved through some genetic linkage analysis. And phosphoproteomics December 3rd is going to be used to look at all these signaling events using some newly collected in vitro samples. And then we also have quite a few prior ex vivo samples that, even though the splenic uptake was very different from one mouse to the next, we have those numbers. So we can always use that to account for the differences that we see and see if we can find some associations maybe in ones that fit within that 1 part per million, just like the Balpsy mouse did. And so, yeah, future grants, we're hoping to look at these 2 distinct arms to identify some of these biomarkers of mercury toxicity and maybe some potential avenues for therapeutic intervention. And so with that, I have to thank everybody involved. Paul, who is sitting right there, Al Rosenspire, Morgan, who is now a medical student here at Wayne State. She was working with some of the early stuff with the in vivo models, and Randy and Rita also were extremely crucial for those studies as well. Paul's got a couple of new postdocs that I listed here in anticipation of December 3rd's activities. And then, as I mentioned, Savannah Hughes, so this was us getting our flu shots the other day. This is Savannah right here. She's been exceptional of just putting up with, what do we have to do to try to get some preliminary data for this to, submit a grant. Maria, my lab manager, helped out a lot with those initial in vivo studies as well. And then everybody else in the lab has had some contribution, if not to this direct project, to the cancer related stuff. And then money drives everything. And so I'm, very grateful to be a part of the CURES grants. And then we also have quite a few other mechanisms that have supported this along the way. And with that, I would be happy to take any questions you might have. Yes.
Heather, is it possible to combine the DO mice with specific gene knockout? Because if we want to consider other contributors to sensitivity to a particular.
Absolutely. Yeah. So you would have to do it as an F1, just like we've done for like the cancer studies. But that is something that when I was a postdoc, in addition to looking at like cancer vaccine response, we were looking at like genetic regulators of like tumor onset. So putting an oncogene in, so you have to have, your genetically engineered mouse model and then cross that with the DO. So in that case, they would only be, F1, but there's still quite a bit of genetic variability in that second chromosome.
But do you have, I'm really not a mouse person, but do you have to cross them with all your DOs? Because you got like lots of different DOs, right?
Yeah.
And that's the strength of the analysis, right?
Yep.
So you have to do a cross.
It does take into account kinship, so it will make sure to account for that as a covariate when it's doing like the strength of association. If you're using a lot of siblings, you're going to end up like within those F1s. If you use a lot of siblings, there's going to be a lot of similarities in there. That in some ways can actually be a strength. It's going to bias towards the loci that you have represented in the mice that you use. But when we've done the F1 crosses, we'll start with like somewhere between 40 and 80 DO mice, breed them to get the F1, and we typically use like 2 siblings per experimental condition.
Okay.
And so most of these mice will produce somewhere between 4 to 10 pups in a litter, so that usually you get enough representation of each DO mom, the DM, in order to be able to like go back and do evaluation.
Yeah, I'm thinking for our epilepsy studies and because the other genetic contributors are a big unknown. And this
Now, until you start crossing with the DO, and then I mean, then the other question, of course, becomes, is it all going to be a mouse specific phenomenon? So that's something that we always get, is that, but again, it's just like, what are we just going to trash all mouse work? And of course, the answer to that is no. And in many of our cases, I always lean on the fact that like the PD-1 and CTLA4, those were all discovered in mice. So
it gives you a clue to go back and look at the human genomes of the individual patients to see if they may or may not have.
yeah, and that concept of like the genetic similarities, synteny. So the fact that like the chromosome 13 locus is a bad example of this because it was just the one gene. But in our other two loci, the entire block of DNA, the genes that are in order on there are the exact same order in humans. It's just on a different chromosome. And in some cases, like SNAX3, 100% amino acid sequence identity to human. So functionally, it's probably the same. But, of course, then you just have to go back and look in the human population. But I think that would be a great example of somewhere, if you were interested. I know the numbers look daunting. It's really not that bad. Maybe I just have a high pain tolerance threshold for these types of things, but it's really not that bad. Especially if whatever you're doing to manipulate or whatever your phenotype you're reading is not something that's like super complicated and takes a lot of time on the part of whoever's going to be doing the measurements. Yeah, I think that would be a good avenue for you if interested.
May I ask a question? Can you hear me?
Yes.
Yeah, Heather, this is a great talk. I have a general question about, how well, I guess, either you or anybody ever look, the overlap between, GWAS identified loci from human versus, the similar, phenotype that being mapped, by using DO model, how well they might be able to overlap to the loci.
Yeah, I think it's going to vary depending on what phenotype you're looking at.
Right.
I haven't specifically looked because most of the questions that we've had so far, at least in the cancer realm, there haven't been GWAS studies done. I mean, the checkpoint inhibitors just got FDA approved about 10 years ago. So we just don't have the numbers to do this in humans. And then I also mentioned, the heterogeneity of the tumor is also going to complicate things. So like these things can be happening in parallel, but yeah, I mean, I would suspect that there would be some similarities, and that would probably be the sweet spot if you have both data sets to just look for where it comes out to be the same. But yeah, I think we don't often have good GWAS studies in humans, which is probably where that limitation is.
Of course. Thanks.
Yeah, it's a good question.
job.
Okay. Well, thank you guys.
Well,